hope you can hear me okay. I do have an accent, but it's not hard to understand. And thank you for the lecture on me, which I was like, really no, I just follow whatever I find interesting, which is more like ADHD. But in terms of overview, my first disclosure is yes, by first training, I'm a veterinarian, and which is why a lot of the work is going to be centered around this particular animal model, is because when I came from Australia, I came through Southeast Asia, worked with some primates, and that's not a common phenotype out there in the science world. And so we're going to be talking mostly about non-human primate studies and human primate studies, because we are phylogenetically almost within 3% of each other. I also want to talk, like she said, a little bit of a lecture on the microbiome, in that what it is and what it is not. And there's a lot of pitfalls out there in how to assess it. And I've been talking just now with Dr. Lark about the pitfalls of extracellular matrix. Whatever your scientific field is, there are problems. We're going to talk about aging. And then in terms of aging, specific disease states that are common with aging. So aging is what I study, but it sounds like we're in a combined thing. So anything to do with sort of health as you age, I'll talk about. And that's primarily metabolic syndrome, overweight, hypertension, diabetes. Those are the three things that we know really are important. So the biggest pitfall is that you think that this thing is going to give you an answer, because the microbiome will not. It is a community, an ever evolving, minute by minute changing thing that is like the vapors of the air that is very hard to nail down. And so it will not provide an answer. It's going to be very different between species that you examine. It's going to be very different based on what you eat, which I think is quite obvious because that's what feeds the bugs. It's going to be different based on geography, which is not just geography as in Colorado versus North Carolina versus Australia, but geography as in my house versus next door versus a caravan down the road. There's also time and individual effects, sample location effects. Now, if I had to survey anyone out here that said, what is the microbiome? They'd probably think of excrement, used many words. But actually the microbiome is everywhere and anywhere, soil, tissue, etc. And so location of where you're actually getting your sample is probably really more informative than anything that we are excreting out. So we're going to talk about disease and medication effects and also how to measure it. And this is my favorite quote by a very, very prominent microbiome scientist who works in the atherosclerosis field called Stanley Hazen. And he says, looking at the bacteria in the feces is like looking at the rubble from the World Trade Center and trying to figure out what was here at this time, at this point. It's almost or actually physically impossible. And the reason it's a good quote is because this guy is the one person that has actually done it. He has found one specific bacterial type that creates something called TMAO, which drives atherosclerosis. And so for him to actually say that you shouldn't do it when he did it, I think is worth listening to. So in terms of defining the microbiome, everybody thinks the microbiome and they think of bacteria. We think of E. coli, Shigella, I'm thinking of pathogens right now, but Staphylococcus, you think of bacteria. But the truth is the microbiome is a lot more than that, which is why the pitfall of trying to figure out what it is. It includes other things. If you don't even know what archaea are, this is the oldest sort of microbiome. This is probably the first thing that was generated when nitrogen was electrified by the lightning that was supposed to have created Earth in the green slime that I don't know anything about, but archaea is the first thing that happened. Then we've got viruses, then we've got helminths, we've got fungus, and we've also got phages. And if anyone hasn't heard of a phage, they will by the end of this talk. So phage is a virus-like thing that infects bacteria. So just as you think of the genome is modified, say, by the methylome or some other process. Phages modify bacteria. So you can't just look at bacteria and know what it's doing. You've got to know what's happening in the bacteria. So this is the level of complexity that we're dealing with, and that all of these microbes sit on the surface of the gut, and we're talking about the gut because it is the largest microbiome that we have, talks to cells, can get through and talk to the cells of the body. And so this is where this host body interface actually happens. And it's the biggest place apart from the skin where we actually are talking to our environment. So again, talking about the bacteria is bacteria is only 58% of everything that is in the gut microbiome. So we're ignoring this massive fraction of things that we don't know what are affecting our cells and tissues. And so I want the pie chart to stay in your head, because when you think that we're doing a great job of understanding microbiome science, we're only dealing with half of what is really there. And because I study aging, geroscience is the new term we've given to the study of aging. And it's really this idea that there is this underlying process in which the sort of homeostasis of the body gets dysregulated. And these are those areas in which we think they are dysregulated. I won't say hallmarks, because that is no longer a term we use in aging, because it is not a hallmark. These are just areas in which things go wrong. But the microbiome affects many of them, including epigenetics, because they create molecules. The microbiome secrete molecules which can then affect the DNA. They can actually damage the DNA just because they're inflammatory. They definitely affect metabolism. So it's estimated in some studies that up to 40% of the calories that you actually absorb are actually microbial nutrients. So if you didn't have those microbes, you wouldn't get that calorie. And that's a big area in obesity, because the obesogenic microbiome might be contributing to the fact that those people might not eat more calories, but they get more calories because the microbes are making them. It also affects adaptation to stress. It may even affect stem cells and regeneration, in that they're sitting very, very close to a whole set of niche stem cells in the gut. So that's the geroscience. And currently, sadly, what we tend to use to study everything are rodents. And so I've already told you my bias is that I love non-human primates. But unfortunately, most people do their work in these rodent models, mice, rats, and there are some big problems. So first of all, they have a cecum that is this big in a rodent. And in a human, our cecum is this big in this body. The cecum is just a bag of bacteria. So in terms of effect size, the rodent's massive cecum is having this huge effect, whereas in humans, the cecum of this size is having a very small effect. And so people are using these models probably not in a relevant way. They also eat their own poop, which we don't in general. There is some fecal oral transmission, but not on a large scale. We try to keep that to a minimum. And what happens when you eat your own poop is that it goes through the stomach. You're actually enriching the upper gastrointestinal system with microbes, which is not what we have. We have a pretty low abundance of microbes up here in the upper GI, the duodenum, the ileum. And that's important because the upper GI has this big, big role in metabolic health regulation. So for those of you who are familiar with obesity sciences, the best thing that we've got going right now is bariatric surgery. As you know, drastic as that sounds, but you're basically cutting out the upper GI. The belief is that a lot of the microbes up in the upper GI are a part of that neuroendocrine signaling. And so the microbes up there might actually have a lot to do with how people process and sense whole body energy. So let's talk about sample size with rodents. So in terms of sample size, you know, people study a lot of rodents in relative species, 20, 30, 40 per group. But if you actually read the small print of NIH in terms of rigor and responsibility, because those mice all live 5 in a cage, that 5 mice should actually be considered 1, because they're all eating each other's poop. They're all inbred. They're actually almost biological replicates of each other, as close as one can get. So when you read a paper and you see that convenient N of 5, they're probably reporting a cage, and it's probably an effect size of just 1. And so again, these rodents are giving us these outcomes, which are probably bigger effects than they truly are. They also eat chow, which is kind of like the most refined form of kashi cereal, which has got no fat, no cholesterol, no added sugars, and pretty much polyunsaturated fatty acid and complex carbohydrates, which is about the most heart healthy diet you can possibly imagine. They also have stem cellness like we do not. So a rodent, if you give it a high fat diet, so you just put it on what most Americans eat, its gut will actually grow 30% longer to cope with it. And we do not do that. If you damage the pancreas of a rodent to try and make it diabetic, it will actually regrow its pancreas and fix itself. So they have this unique regenerative capacity that we have lost in evolution. And again, it means that in a rodent, you can fix a lot of things that you can't in people. And so this has led to what has called the rigor and reproducibility crisis. And as you hear more and more about rigor, it's because none of these rodents have translated into science or things that are relative to human health. And so right now, the sort of aggregate estimate is that 80%, 86 or 90% of evidence coming from rodents, when you put it up the evolutionary chain, dies. And we don't get the drugs, we don't get the things. And so species is problem. So that's why I love my non-human primates. I am mother to about 500 of them. My work is entirely non-terminal, just in case you wanted to know, because I'm interested in aging. And so I'm watching my animals age to the end of their natural lifespan. And so I might have some girls that are now at 30 years old, which is older than most people. So old non-whale primates, you can see their microbiome looks very similar to a human, in that most of it, this is the bacterial microbiome, is mostly Bacteroides and Firmicutes, which are these two great big phylum everybody will talk about. And so they have these similar enterotypes, which is a concept that came out of dietary patterns, meat eaters versus vegetarians. They show that similar enterotype, but it's not related to diet. Sorry about that. I don't know who is calling. Ignore it. And then we're going to look at some factors that define the microbiome. And the big ones I want to focus on are diet, location, and disease. And we're going to focus on these factors relative to aging biosciences. So the first one, if you know anything about aging, is we're all looking for a drug that's going to slow aging. And right now, the biggest drug out there that everybody wants to talk about is something called metformin. So metformin is a drug that's treated for type 2 diabetes. It's the most commonly prescribed type 2 diabetic drug out there. It's very cheap, very accessible, and it works very, very well. But it also has big effects on the microbiome. It has great effects on cellular energetics as well. It's got multi-level effects. But the thing I wanted to point out in this, because this was actually a very good study that was done in over 1,000 some subjects, is that the concept was that the treatment with metformin normalized the microbiome. And so what they mean by normalized is it looked more like a non-diabetic control. But the drug effect actually shifted the microbiome in a whole different direction. And it was more of an effect than it was just having diabetes itself. And so the red dots at the bottom are those that are treated with metformin. And you can see they're quite different to the drops that are yellow, which is a diabetic treated with metformin. And they've actually separated in two different ways. And so when you're understanding human disease, humans are a combination of their environment, their age, the drugs they're taking, the drugs they've taken in the past, the diet they took in the past, the diet they're going to take in the future. And that metformin, although it worked, affected the microbiome, did not necessarily make them a normal non-diabetic control. It just made them different and healthier. And some of the things that went up actually were not necessarily good, like Escherichia or E. coli. So speaking of diet, this is probably the leading group that's doing age-related microbiome work. They're out of the University of Cork in Ireland. Very groovy people. They tracked a bunch of people that were healthy and aging going through basically the end of their life. So whether they're living in the community as older people, whether they came in for a day surgery just to get their knee replacement, whether they went into rehab after their knee replacement, or they were in a nursing home, and figured out that on the top, the red green is where they lived and the different sorts of diets they had. Healthiest being that they had fruits and vegetables, everything. Meat and sugar was added in there as diet group 2. Then they had some that ate potatoes, and because it's Ireland, they ate more potatoes. And then diet group 4, they ate only potatoes and some fried fish because they're Catholic and they ate that every Friday. And you can see that on top, the groups classify where they live dictated what they ate. And then as their diet got less healthy, the speckling indicates how much microbiome diversity they have. So as the diversity in their diet went down, the diversity in their microbiome went down. And I think we all understand the idea of diversity is probably a good thing, a robust thing. You want diversity because it tends to allow for more interactions and stability. So this was a really nice example. And then they actually tracked these microbiomes and found that there are a number of different shifts that one could get to end up in frailty. But this is what happens in aging, is that people change their lifestyle. Their bodies are changing, their lifestyles are changing all at once, which makes it very, very hard to discern what's a diet effect, what's an aging effect, what was a medication effect, because all these people in hospital were getting drugs as well, which is why the primates provide such a nice counterpart. So here are some diet studies in primates. On the left, I was very interested in high fructose corn syrup. So we were treating animals with sort of human or teenage relevant high fructose corn syrup. I gave it 5 weeks, just drinking Coke. This was not long enough for them to shift their microbiome. So as much as we think the diet might affect things, you know, if you're going to do things, you know, a 6-week ketogenic diet to lose weight is not going to permanently shift your microbiome, probably. But this plot on the right, this was 3 months. This was an American diet, sort of sugar, fat, cholesterol. 3 months was long enough for them to separate out. So this stability is there, but it's not that stable. It takes a little while to reshift patterns. And this is important in terms of any study design, particularly in rodents, and where rodent studies tend to be done over the course of weeks, maybe months if you're lucky. So diet will do it, but it takes some time. But it doesn't just change the microbiome in your gut, it actually changed the microbiome everywhere. So this was a study that we did in non-human primates. They were fed a Mediterranean type diet versus a Western sort of hamburger diet for years. And we actually looked at their breast microbiome because we were looking at differences in cancer risk, which is known to be lower with the Mediterranean diet. And you can see on the graph on the left, this is just PCA plot, that it's separated very nicely. Again, we're just talking about the bacteriome. And then over here is some of the specific bacteria that did change in response to the microbiome. In response to the Mediterranean diet. And I've boxed out there one that everybody knows is lactobacillus, because lactobacillus is, you know, your yogurt. It's all good. Yogurt is always good. I don't disagree. I do promote yogurt. All of them turn up positive in pretty much every study, so you can't go wrong. But you see different shifts in this tissue microbiome, and this is unrelated to what's happening in the gut. This is actually your tissues telling the body what microbiome it wants to have, based on whether it is translocated from the gut or whether the tissue itself is in different sort of nutrient or inflammatory status and selecting what bacteria wants to live in it. We're not really sure. But diet will change, not just in your gut, it will change everywhere. And then this is actually the same data set overlaid with the virome, so that other big component of the microbiome, showing just, all I want you to see is how complex it becomes. We've been looking at the bacteriome as part of the microbiome because it's probably the easiest thing to access. We do 16S sequencing, you can map almost all of them, but that doesn't account for the other 50%, which is phages, helminths, blah, blah, blah. This is just the viromes. This is just adding 25% of the complexity on it, showing how much more interactive it becomes and how the changes go up and down with the Mediterranean diet. So huge area of research and still very, very undiscovered. So geographic effects, the microbiome doesn't just live in one place. So we can have small or large changes. And the plot on the left is actually the same data showing a few things back by fructose versus non-fructose exposure. But this is now by who lives with whom. The colors are basically, my primates were living in condominiums, we call with they were two to a house. So they were shared like couples. And the colors clustered together because you share the microbiome with those that you live with. So your family will share your microbiome, your roommates will share your microbiome more than you will with someone that lives 2 doors down. This plot here on the right, this is human data measured over 3 months, showing exactly the same thing, that people are pretty stable, which is why you have to feed somebody something a long time for things to shift. People retain their microbiome pretty well for months at a time. So you have to perturb things pretty well to get things to shift. And this has been shown over and over again. And this is again why mice in a cage is all one unit. They are sharing a microbiome. And there is some, this middle graph here is also data from the group in Cork, Ireland, and they've taken their research into a really interesting direction that I'll just mention, because they've been looking at a group of gypsies called the Irish Travellers. And so these Irish Travellers are, they're like Roma, they live in caravans, they have lots of dogs and horses, and they travel around and they're really tightly packed. And they look at their microbiome versus other Irish people that I showed in those diet groups before, healthy diets. But the travelers, with their interaction of all the dirt and all the dogs, was much healthier and more diverse, and just as stable. And so they're actually making a case for the fact of kind of like the hygiene hypothesis, that we have cleansed ourselves and we've actually hurt our own microbiomes. And so I just think that's kind of a neat idea. But the geographic effect can be very, very large. So this is a big study of thousands of people grouped. I can't even remember what the intervention was here. But the point I wanted to point out here over on the right is these are countries. We've got Italy, UK and France look pretty much the same. Poland and Netherlands, who eat pork and potatoes, they're over here. They eat the same. They're more closer together, sort of physiologically, I mean, geographically as well. UK and France are obviously pretty close together. Italy, surprising they're not more like the French, but they're not. But the separation by country is so much bigger than the separation by your intervention. So these effects are dominating. So if you're ever going to do some sort of interventional trial, you're going to find that there are other factors at play that are going to be so much bigger than the factor of whatever intervention it is, whether it is giving a probiotic or making them exercise 3 times a week. The geographic effect is something you have to model in there because it's so much bigger. Again, the R squared 0.33 by where you live versus the treatment effect, which was 0.08. And this is repeated by another very high profile publication that came out a while back now in Nature. This is in their supplementary data, but they actually listed out the effects, which I thought was transparent of them in terms of the treatment, 3.2% of the variation, disease a little bit. The country where you came from was 3 times more important about what your microbiome looked like. And then they all interacted significantly. So again, just adding complexity to understanding the microbiome and why the microbiome is not going to provide an easy answer. There are some actually really, really interesting studies where they've gone in Europe, in Switzerland, yeah. And they literally, one family here, they went over the mountain to the next family, totally different. And they had ostensibly almost the same environment. But geography can get even smaller than that. And so here's where I want to sort of pivot into niche microbiomes, which might be more important than the stuff that's just being pooped out, because that's the stuff that our bodies are not keeping, right? It's coming in and it's going out. Feces has a very different microbiome to what's actually sitting on the surface of the mucosa. So this is your intestinal lining. So the surface of those intestinal epithelial cells is where the microbiome is actually talking to cells and the immune system. What's sitting in the lumen or the feces, again, is in a separate containment zone, literally a tube that's going on its way out. So what we really want to know is what is in the tissues or what is talking to our tissues. And so we've been trying to focus a little bit more on what microbiome is doing at the mucosal level in the gut. And so that makes us want to talk about the mucosal barrier, because this is what keeps those microbes from being in the tube versus in our tissue. And so the mucosal barrier is this really important thing. And so when I talk about biological aging, I just want to bring, this is a completely unrelated plot, but this is talking about how walking speed predicts how long you live. And this is hugely famous. Like Stephanie Studensky did the most beautiful job of showing women walk faster than men. That's take-home message. And we live longer because of it. I think this is great. So, but basically at any given age, if you can figure out how fast you walk, you can predict how long you're going to live, which is kind of terrifying. And I know you're all going to go tearing out of here at like as fast as you can walk. And I think about it all the time. But the interesting thing about walking speed is that, you know, as exercise scientists, as a lot of you are, you know that brings so many components together. It is bioenergetics, it is the muscle strength, it is joint health, it is balance, it is neurological control of the tissue. It is this sort of composite measure of how your body is functioning at that level. And so I think it's a great biomarker. It's a sort of biomarker we should all look at, which is why I think the mucosal barrier is also something like that, in that it comprises multi-system organization, in that it's got the microbiome, again, the lumen, stuff that's leaving. Then it's got mucus, it's got this microbiome that's then sitting on the surface, which is actually talking to cells, which have immune cell components, inflammatory cells, it's got stem cells, and it's got blood. So it's kind of this barrier function. I think of it as a one-stop shop for how this whole tissue is functioning. And so looking at mucosal barrier function is my sort of one-stop, how is our bodies functioning? So here's some data just to show that the bacteria sitting on the surface are different to the bacteria sitting on the lumen. This was done quite a while ago now, but just a good old everyday gastroenterologist doing routine colon polyp screening. We started taking some biopsies and just looking at the mucus versus the actual cells. And you can see there is a different separation. And we've done this too. Again, over here. And I can tell you that I've looked for microbiome differences in aging in our monkeys over and over again, and you never see it. But when you look at the mucosa, you will. So the mucosa over here is enriched in firmicutes, which is the light blue color. These are the better bacteria. These are the ones that we like to think of that should be there, that are happy. And that makes sense. So you want beneficial, more gram-positive bacteria sitting on the cell surface. So it makes sense that it's shift. It's a selective mucosal niche. And so this mucosal barrier, it's very, very conserved. And so that like walking speed, I think of it as being this one biometric measure of how multi-systems are functioning. You see aging flies, their barrier decreases. And I think I've got a video embedded in here, we'll see. So basically you feed flies fluorescent dye because they just eat it. And as they get older, their guts deteriorate and that fluorescent dye starts circulating as they get older, and then they die. They actually die very repeatedly. As soon as they start going green, they'll be dead in 3 days. I mean, that could be terrifying for us. Rats, they get older, their mucosal barrier decreases, and it's measured by this thing called lipopolysaccharide binding protein 1. And I'll use that LBP as a marker in data going forward. Zebrafish, proportion of zebrafish that turned fluorescent positive, we just put the dye in their water. As soon as they turn positive, they start dying. And in the primates, we see it all the time. As soon as they get old, their barrier marker LBP1 increases significantly. And again, not just the mucosa, this is also seen in the brain. There is a brain microbiome. There's actually viruses in the brain, not just bacteria. And it's the same thing. The brain has a blood-brain barrier very similar to the mucosal barrier, actually even more simple, that is keeping bacteria out. And so its function, again, would be another biomarker of how well these systems are functioning. So the barrier, I think, is a really key indicator of how global systems are functioning. And this happens in obesity too. They call it metabolic endotoxemia, which is just a general term for having more gram-negative lipopolysaccharide in circulation. And so this is an inflammatory signal. So lipopolysaccharide is the wall of a gram-negative bacteria. It's a negative thing. You don't want bacterial bits floating around in your circulation. It sets off inflammation in all tissues. And so in obesity, we're finding that there is more endotoxin circulating. We don't know if it's because their barrier is leaky, if fatty acids, if it's saturated fatty acids, which are most of your animal fats, it can mimic endotoxin and set it off. But again, everything relates back to whether this inflammatory response comes to a microbial signature. So we see this in people. In aging, we see that LBP1 goes up, and it's associated with all the things that you expect to be with age in terms of inflammation. And it's actually also related to their physical function, that walking speed. And so if you actually associate the walk test with the barrier function, it is significant even when you adjust for all other factors. So again, this is why I think that the barrier function is this one-stop shop for understanding gut health. So a number of studies have actually linked the fecal microbiomes to frailty, but nobody can tease out the environment from that frailty functionality. So frailty is this idea that the physical function, immune function, everything's declining with aging all at once. The microbiome also changes at the same time, but there is multiple different sorts of microbiomes that can get you to a frail space. So there isn't really one spot. So we have other primate models. So we want to look at this microbiome barrier function. And one of the best models actually surprisingly out there is HIV. So this is an immune compromised state. An AIDS patient actually looks like an accelerated aging patient. They have heart disease, they have all sorts of other comorbidities. And what is interesting with these guys is that when they become infected, they actually become endotoxic. So their microbiome is able to translocate more readily. Now in the monkey world, because as you might have known or may not have known, HIV originated from primates. There's all sorts of horrible stories about how people think it started, but it started in the monkey population. So there's actually a population of monkeys that can get HIV but never become sick. They never develop AIDS. And so what's interesting is they call these controllers or progressors. And so the monkeys that are able to control their HIV, they never develop endotoxemia. Their intestinal barrier remains strong and intact. And this on the right, this sooty mangabe monkey, this is one of these monkeys that are able to basically resist HIV. They never show any problems with their microbiome barrier. It stays intact the entire time. And so I just wanted to use this as an example of how important that function of that mucosal barrier is. So in our old monkeys, we see that the mucosal microbiome shifts with age. This is what they look like when they get old. They get all sort of droopy and doddery, but they're adorable nonetheless. They actually show a change in their mucosal microbiome, shown on the right, which is not reflected in the fecal microbiome. If I haven't said it enough time, that's what we're pooping out. Do we really care? This changes, and here is what's called diversity index, which is just a measure of how many different species are represented in a sample. So counterintuitively, we usually think diversity is a good thing. In the old monkeys, they have high diversity, and that to me is indicative that their barrier is unable to keep the niche balanced the way it should be. It's allowing more things in there than there should be. It's supposed to be an anaerobic environment with a lot of mucus, slightly acidic, designed for a certain set of bacteria. When we get old, that sort of specificity is lost. And so this makes me want to talk about other common diseases, because aging is a common disease, but there's also lots of other common diseases like diabetes and hypertension. So here, this is an example of hypertension. And so we took mucosal microbiomes from hypertensive animals versus non-hypertensive animals. And we can see that their mucosal microbiome is disrupted too. And again, I'm still just talking about the bacteriome at this point, but nothing else is different. Environments are the same, ages are the same, weights are the same, they're on no medication. But the fact that their barrier is changed by the fact that there is changed perfusion due to their blood pressure, they've already disrupted that niche and that microbiome has changed. So metabolic phenotypes, hypertension is part of something called the metabolic syndrome, which is obesity, cardiovascular disease, and hypertension and dyslipidemia all rolled up into one big package. We see that happens mostly in people that are overweight. So this is the red on the bottom, what we call metabolically unhealthy obese. But there's actually 15% of lean people that are unhealthy as well. You can't tell from the outside. And there's actually a third of people who are overweight that are perfectly healthy too. And, unfortunately have to live with a lot of stigma, and that's probably not fair. The vast majority of lean people are healthy. But there are these 4 phenotypes, and we see them in our monkeys at exactly the same rates that we see in people. In the obese monkeys, about a third of them are perfectly healthy. In the normal weight monkeys, about 12% of them are still unhealthy. And these are the risk factor prevalences. They have 20% of them have high waist circumference, which is obese. high glucoses, triglycerides, blood pressure, and HDL. So we were interested in what the microbiome was doing in these categories, because there's a lot of controversy. The fact that you can be healthy and obese is something we don't really want to talk about, because our whole mindset is geared towards lose weight, be a normal weight, be healthy. But if anything has shown us in the obesity epidemic is we're not losing weight, there is nothing that is working to lose weight. Even bariatric surgery doesn't allow for full weight loss, and that we're continuing to becoming more overweight. So for me, the question is, what is the role of the GI in this? And is there a better way to stay healthy when you're obese? Currently, most people who are healthy and obese, about 50% of them will stay that way after 10 years, and 50% of them will eventually become healthy, which isn't bad odds if you're a betting person. So there is a controversy. What do we need to do with these healthy obese people, or what is different about them? And obesity is typically defined as diabetes. So some people, including me, have started to look at the tissue microbiome. Again, this is a niche. And we do have bacteria in all parts of our bodies. And so there are really no difference by disease status in a diabetic per se. But over here, they found some very interesting associations with the bacteria type in the omental fat, which is in here, and inflammation. And so the omental fat is interesting because it's sitting around our intestines. And so the bacteria theoretically could be walking right across into the tissue. And so what we found in this was exactly the same thing when we looked at omental fat by what's called healthy, whether they're lean or obese, which is the green and blue, versus unhealthy, which is lean, which is red and blue, darker blue. They're quite different bacteria in the tissue niche. So the fecal microbiome, again, I'm going to show you, was entirely the same. So it's not what they're pooping out, it's what they're allowing to live in their tissue that is different. They are quite distinct. So the fecal microbiome is clustering over here, and the tissue microbiome is clustering over there, which is really asking bigger questions about why does the body allow certain bacteria to live where they do, and how that differs by disease state. And if we get into the bacteria, what was clearly different in these unhealthy groups on the side was that they're more gram-negative. If you just break it down very easily, gram-negatives tend to be the bad guys, gram-positives tend to be the good guys, unless you're a methicillin-resistant staph. But in general, your lactobacillus, your bifidobacteriums, or your yogurt is in the gram-positives. And so what we found was this visceral adipose had a whole bunch more gram-negatives sitting in there. And these gram-negatives actually did, the phylum and certain genus did relate to how much local inflammation, not circulating inflammation, but local tissue inflammation we saw. And again, these are both more gram-positives have lower inflammation. So why those beneficial microbiomes come and go, we still don't know, but it's important to realize that they're there. Whereas in the gram, if you look at metabolic syndrome score, which is just basically how unhealthy they are, the more gram-negatives you have, the worse your score was. And if you know clinical science or monkey science, anything that's got a correlation of over a 0.5 or 6 is actually really strong, because there's so much variability in people. These are actually pretty strong associations. So on the bottom, again, all your bacteria that are in your yogurt, bifidobacterium, lactobacillus, lactococcus, these are all the probiotics that you buy off the shelf. The more of them, the healthier you are. So, we're seeing patterns, and that's also a big key pattern of healthy diets, whether it's fermented Korean kimchi versus fermented yogurt versus other fermented things, tofu. That is part of a sort of this healthy idea of what we've got going on. So I think, this is an interesting picture and really drives us to look more at niche microbiomes as a way to understand health, not at the fecal. But what I get confused with, and I think everyone else is this: does the microbiome drive the health, or does your health allow microbiomes to survive? And that is a question no one's really quite figured out. We have some indications that the microbiome might become before the host. And these are the key areas in where this is kind of pretty clear. Ulcerative colitis is microbiome driven. C. diff, obviously microbiome driven. Neonatal programming, there's a lot about C-section babies being different. Their immune systems are different. They don't have the same microbiome when they start life. Babies with necrotizing enterocolitis. These are clear indications of it's happening to the baby first. And we have some evidence for this. These are some of my monkey mothers out there. Their microbiome, if they're obese versus normal weight, is less diverse. That's well known in humans. And if you look at their babies, this is measured in the first 2 or 3 days of life, the baby's microbiome is following the same trend. So they are inheriting this microbiome, and that is well understood. And that inherited microbiome is probably training their immune system, which could then future train what bugs are allowed to live in the tissues. And so that would suggest perhaps microbiome before host. But then let's look at another situation where the host might be driving the microbiome. So again, I'm interested in hypertension. In this study, I had this cohort of monkeys that were, some of them were hypertensive and some of them weren't. And I looked at them for a couple of years because you have to look at everything for a couple of years for it to really make sense. Microbiome takes years, just remember that. So these guys were either hypertensive and decently hypertensive. AHA is considered 140 millimeters per mercury hypertensive over a couple of years. And what we saw is that at the point of hypertension, when they're already hypertension, they already had leaky gut. And that as time passed, that leakiness got worse and worse, almost exponentially. So that hypertension seemed to be driving a real problem at that mucosal barrier. And we've already know that the microbes living on that mucosal surface are different. So we looked at the microbiome, and it really didn't change on the global level too much. But if you started to look at some of the big players that we all know, family of Lactobacilliaceae, this is a happy microbiome microbe. When you're hypertensive, the blue line, it gets worse over time, but it took years for it to drive down, which suggests that the body is driving an eventual microbiome. So this is a host before microbiome. And ruminococci, which is a bad player, it went up, but it took years for it to actually show itself. But this is fecal. Maybe things would be showing up earlier at tissue level. And it's probably easier to appreciate this by PCA plots, where you can see it's all mishmashed at the beginning. They're starting to separate at about a year. By 2 years, we're different. So to me, this is an example of why you look long and why you have a host driving a fecal microbiome effect. So the other thing about host before microbiome is looking at how these microbes are interacting with each other. And so this is a new metric that I'm trying to promote in terms of network science, in that these guys don't live in a silo. They're not this bacteria living with this bacteria, because this bacteria is actually secreting stuff, infecting with phages that guy. They're talking to each other. They live together. And so you can see on the left, the mucosal community, this is again tissue, niche level, is robust. They're talking to each other. They have a lot of interactions. Green, it's a positive. They help each other survive. Red lines means they're less likely to survive together. Whereas an old mucosal community is disconnected and basically just not as much interaction going on. And this goes along with the fact that we actually had a more diverse set of microbes in the mucosa of these old monkeys. So they're probably never meant to live together and talk to each other. They haven't developed a community. So we have a disjointed and more diverse community living in the aged gut, which is probably not a healthy thing. And again, that's the diversity plot, just reminding you there are more different sorts. And I think these sorts of metrics of networking, however we get to it, because it gets more and more confusing, is a good way to start drilling down into what sort of things are going on a global level. And so we just did this by how many nodes there were, which is how many bacterial species there was an interaction, which was the same. But in the old monkeys, they had far less talking to each other, co-occurrences or co-exclusions. And so I think this is a nice, simple way to show that the network has deteriorated as we age. So it's a novel metric, but it gets worse because you can get down. That was a family level. This is the genus level. And it can go down one more level too if you actually do whole genome sequencing. But you can see the young, just on a visual appreciation, you can see there's a little bit more green. People are actually talking to each other and like happy to help each other live, whereas in the old community, we're a lot more red. which is co-exclusion. We don't like each other, don't want to live with you, don't know why we're here, which might be the host actually having a poor niche for them to live in. But so this is sort of the metrics where I think that the work needs to move to, because focusing on an individual species is not likely to get us anywhere. So I'm going to talk about a different model. This is called aging and resilience. So resilience is this concept that when you're older, you can have a stressor. Let's just say you go in for your knee replacement surgery, and a person who's aged well will just get back up out of bed. The physical therapist will get them going and they'll walk out the door. Whereas someone who's frail spends 4 days in bed, doesn't get up, never retains muscle strength, and they never come back. So they don't have resilience. We have this very extreme model of looking at resilience. So in human medicine, they'll look at bed rest, a procedure, they'll challenge them with a diet, and we do radiation exposure. Radiation exposure, which sounds really, really rough, but radiation creates an instant DNA damage, an instant inflammatory response. It actually damages progenitor cells, does a lot of things, but it ends up after you're recovering having a lot of comorbidities that look like aging. They have heart disease, they have diabetes. So we use radiation exposure as this sort of acute stress that accelerates an aging-like phenotype. And these animals also have reduced robustness, and robustness is a different concept to resilience in the aging world, whereas robustness is you had the hit, but you didn't go down as far, right? So the person who had to stay in bed 4 days, you only stayed in bed 1. So you can be robust and resilient, but you can't be resilient without robustness. So in radiation, we actually went an extra step and did the whole genome sequencing so we could get to the viruses and all of the phages and all the other things, just to look at what was going on. So here is, again, the picture of what happens when you get the whole thing there. You've got all 58% of bacteria, but you've also got the viruses. When we look at survivorship versus some animals don't survive, that would be really not resilient nor robust. They did not survive radiation. There was actually really nothing specifically different about their pre-irradiation microbiome. And this is based on work that has been done in rodents, where rodents had their microbiome, fecal microbiome assessed, and some who had a certain profile could actually make it through radiation, no problems, and some didn't. So we wanted to replicate that. And what we saw was that the whole microbiome, if you looked at it as a big thing, really wasn't different in terms of the species, but when you look at how they clustered, there was a very big difference. And we only had 3 that didn't make it through the study protocol. This is not my study, by the way, but those 3 were very different. And this is before they had their stressor. So they started out in a place that made them vulnerable to not surviving. And what was different was not their bacteria. So that 58% of their microbiome was not bacteria. It was very different in a different space. There were a couple of bacterial species that were higher, and I'm running out of time, so I'm just going to point out that the species that were higher in survivors, these are the blue bars, were again all your yogurts, all your bacteria that you would find in your probiotics over the counter, but also this super cool dude up here, and I'm going to point out fecal bacterium presnitzii, can't say it, because this is the one that's come out in the lay press as being one that goes up with exercise, being that you guys are in the health and exercise science. They did an exercise study and they found this species increased. But if you have this before you go into radiation, i.e. you exercise before you go in for your radiation therapy, this is a good player. This will help you get through it. But again, all of these were not unexpected. These are healthy bacteria we should have. They did make you more resilient. Ones that were upregulated in those that didn't make it were all gram negatives, bad dudes, E. coli, Klebsiella. These are pathobionts. So this is also not surprising. But what was really interesting was about the virome. Virome was different. We definitely see that there is a difference in the virome, and most of it was actually not viruses. These are the sort of viruses. It was actually just one. It was actually phages. So I'm going to skip over it because I am running out of time. But the phages were really what made the difference between a healthy and an unhealthy microbiome. And lastly, because I know cellular senescence, if you are in the aging field, you will know about cellular senescence. This is the Hayflick idea that basically you can only replicate so many times before you senesce and no longer keep going. This is basically cells that they're zombie cells. They stop dividing, they stop replicating, but they're not dead. And they also secrete a lot of inflammatory factors, and it increases as we age. So if you're into this field of aging, you will know that metformin was old school. We are now doing senolytic drugs. This is what's going to help you age healthily. And the biggest one, quercetin, there was actually a scientist out here that was doing a lot of flavonoid work, and I've forgotten his name, but it's a flavonoid. You can buy it at Walmart. You can buy it anywhere, bright yellow. You can take it. Dasatinib, unfortunately, you can't. It's a chemotherapeutic. And there are a number of trials going on because we believe that the combination of these removes senescent cells and slows the aging process, or is the theory. So we decided to do a trial in monkeys that were older, unhealthy, obese. A lot of them were diabetic. We gave them 5 months of these drugs. It's only once a month, which is attractive. And then at the end, we added in caloric restriction because we all know that caloric restriction extends life, makes you healthier. And we just wanted to see what the interaction was. So what was interesting in terms of host versus microbiome is we did mucosal biopsies, and it was not different. So here we have a drug that did not affect the microbiome. However, it did affect the mucosal barrier. So we have, this is our marker of LBP, and it's broken down, it's all adjusted for baseline and whatnot. And at the end of the study, those who were treated did have a more robust barrier of their gut. And that actually got even better when you added a little bit of caloric restriction onto it. And it was only a little bit of caloric restriction, 10%. And then if any of you know aging, that's nothing. Aging people want to caloric restrict 30 or 40%. So this was just a mild diet, they were fine. But we did see that. We also saw that caloric restriction reduced IgA in circulation, which is also a measure of how much microbes are going through, and IgA, which is secreted to combat that. And so in end, I just want to, because I know I'm totally running out of time, is that there isn't an aged microbiome. There are a couple of species that will come up over and over again. Lactospiraceae, centenarians have it. The fecal bacteria Presnutzii is going to come up over and over again, but that doesn't define a microbiome. It's just one species out of thousands. So I talk about centenarians because I really do think they are very interesting. Mostly they have diabetes rates of a 50-year-old. They're protected, but they have heart disease. They have high blood pressure. They have all these other diseases, but they don't have diabetes, and they do make it to 100. They have fecal species that, again, more gram-positives and a couple of those species that I've mentioned a few times, Lactobacillus, Lactococcus. And then, you know, our survivors of radiation show that this is probably a real phenome, even in a clinical setting. So there are some attributes to a fecal microbiome that might show age-relate positivity, but it's not a definition. So take-home message is that, you know, animal models are great. They can define, you can control everything, but don't use a rodent. You can't get the power, you just can't, and you can't get intra-individual sampling. And as the microbiome is stable, remember, months to years, you can't sample rodents for that long. People don't do it. Definitely intra-individual sampling is the way to go. Make sure that you have microbiome understanding that host versus microbiome, you don't know which one is coming first or second. So make sure you get basal samples in anything that you do. And that we definitely need to move towards whole genome sequencing, because so far we've only been looking at 58% of the microbiome. The viruses and the phages are interacting and changing those microbes in a way that we haven't even started to look at. And so we need to understand how to metricize that interaction rather than try and pinpoint certain good players and bad players. And more recently, you know, the fact that the microbiome is actually evolving within the host. So just like people, we accrue mutations in our genome as we get older, exposed to the sun. The microbiome is actually mutating its own DNA at the same time. And so it's changing within the host. And so those changes might be different with disease. So looking at the microbiome genome within the host over time is also important. And then again, these things that we're using right now, prebiotics, synbiotics, which are like your fibers, eating more dietary fiber, inulin, these are still rational scientific approaches because they do promote positive, gram-positive bacteria survivor. The fecal bacteria Plasmatiae, that player that looks like it's going to work out, it makes butyrate, which supports in the cells of the mucosal barrier. Still a rational scientific approach, but it's not going to help us define what the microbiome is. So I just, I caution people when they go into the field. It does look like that. It looks like that. And so don't try and pin yourself on one microbiome. I think you'd be a much more, don't pin yourself on the fecal microbiome. I think you have a much more healthier sort of research program going forward if you pick a disease or a tissue and try and understand over time what's happening within that certain space, because it's probably going to provide more targeted information. And so I love these guys. They've taught me so much. Lots of funding from different places, lots of questions. There's not a whole lot of time, but I answer email all day, and I answer within 15 minutes when I'm enthusiastic about it. And also I have to tell that we are looking for postdocs. We're looking for 3 right now. Clinical trials, microbiome science, and also aging and microbiome. So all I can say is Wake Forest is a really wonderful place to live and work with these really great guys. And if you're into the outdoors, it doesn't suck either. I know you guys think you have a lockdown on mountains. We've got old mountains, the Appalachians, but Louise can say, we get out. And it's basically free to live. It is. But yeah, so I would love to talk to anyone who's in their last year of their PhD or whatnot, because we have so many fun projects going on that I haven't even started to talk about.
Probably only have time for 1 or 2 burning questions.
Throw it out there. I am very informal.
Prioritize students.
Oh yes. The microbiome in the brain is certainly people are looking at, because they're interested in whether the glial cell activation is related to local microbes. I have looked for microbes in CSF and I have not found them. And I believe that actually, because I think they're probably in the tissue. So I think if we actually start looking at brain, we will find them. And again, it will be that interaction of whether the local host immune milieu or the vascularity or whatever's happening, whether it's creating a niche that supports good or bad bacteria. I think that's a hot area, because I don't think we all understand there is no sterile space in the body anymore. Like that started when they found bacteria in the placenta. Huge argument. There are no bacteria in the placenta. Yes, there are bacteria in the placenta. They're everywhere. And so I think understanding how each tissue is reacting to that particular microbiome is going to define how we think about disease going forward. How we change it, however, I don't know if that's going to change drug design or development, but it at least gives us another way to look at how these diseases are forming. But so yes, I think it's there. I have not found it in CSF, but I am quite sure that if I could find it in brains, but these guys are all alive, so I can't do it. And I'm not doing a brain biopsy anytime soon. I'm sure it's there. Same with the pancreas, which is also a neuroendocrine cell, so embryologically derived the same way. A lot of people think type 1 diabetes and even type 2 is related to the local microbiome, local viral components. We could start looking there. Any other questions? All right, everyone's going to run out of here and grab yogurt, right? That's what you should do. Like walk as fast as you can. Because yes, yeah. I mean, this is all great, but lifestyle is still the best thing we've got right now to modify risk for disease. So I can't say that enough. I wish NIH would actually fund public health sciences more than they fund some of the stuff we do. Yeah. Yeah, that's a really there is a difference. This is what they call enterotypes. It's been not as stable as people wanted to believe, but again, they've based it on fecal microbiomes, which is entirely what is the bacteria feeding in the lumen, which makes sense because vegetarians have all this fiber and they're eating it, and so the bacteria is the same. Whether it's different at the niche level, I don't know. And I would guess it's probably not as big as the difference as we see in fecal microbiome, but there are differences. But there's a lot of controversy because the healthy microbiome in European countries where meat is still, quite a big part of the diet, and they're just as healthy. There is no healthy microbiome. You can have high fat eaters, high this eaters. We were talking about blue zones earlier. Those extraordinarily long-lived people eat completely different things. I mean, you've got the Hanzu Peruvian jungle people, and then you've got the people in Nairobi, and then you've got Okinawans, and then the Sardinians, and they all eat this entirely different set of foods. So I really think it's more of a case of the host is able to somehow retain the right microbiome in the right places. So there is a difference. You can find it. Whether you can transplant that and end up with a healthier person, I don't know. And that's a whole nother question we were talking about. We're transplanting fecal microbiomes, which is unlikely doesn't persist. We know that at least. People revert back to their natural microbiome in a course of 6 to 8 weeks after the treatment. So yeah, I don't think that's going to be the way we go in the future. I think more likely the extracellular vesicle transplant is probably going to be more viable. So yes, difference, but I'm not sure that it explains the differences in health.
So walk quickly out of here, eat yogurt, do not get a fecal transplant.
Yeah, no. Well, you don't know what viruses you get.